about 7 years ago, I got an email from a young researcher from Spain who was interested in complex systems and the brain. And so I got this email from a young guy in Spain, interested in brain and complexity, inquiring about a postdoctoral fellowship. And I was very quick in responding. Dear Joaquin, great CV, but I have no funding. Good luck. That was literally the length of my email. And happily, Joaquin persisted and sent a follow-up email. And I managed to get 1 year of funding for a postdoc shortly thereafter. And I would say the rest is history. Joaquin came in 2011 and ushered in what I still call the golden period in my lab, with Joaquin arriving two graduate students, Andrea Wiener-Königsberger and Rick Betzel, who you'll see in the talk. And that was one of the most exciting, productive, creative parts in my life, and I'm very thankful for that. And I'm also very happy to see that Joaquin has found a good spot for his next stage in his career, and I'm very happy that he's here at Purdue Brain Networks. I don't use the term network in a metaphorical sense. I mean it literally as a mathematical object of a collection of nodes and edges. It's a very common description of complex systems, such as the brain, systems that can be broken down into elements and their interactions. Network science has made a lot of inroads in other disciplines, from social sciences to technology to biology. Examples here, social networks, Twitter, blog networks, and interactions among proteins in a cell. And there's now a new area, I think the last one, where networks have made a difference, and that's neuroscience. And that's the science now of brain networks. Brain networks have a lot of history now behind them, about 10 years, 20 years of research. And I want to give you a little bit of an overview in the beginning. What do I mean by brain networks? What are they? Before turning over to topics, networks across different species, including the human, functional connectivity, and I'll end with a little outlook. This is a diagram that may seem familiar to many of you who have studied computational neuroscience. It's actually deliberately built on a graph 25 years ago now from Terry Sinofsky's work. We recently had this in a review article, and it's supposed to show that we can actually acquire network data in neurobiological systems across very many different levels of scale and in both time and space. On the extreme lower left-hand corner of this diagram, you see molecular networks, interactions among molecules that make up cellular systems. Upper right-hand corner of this diagram, we have interactions among individuals, organisms in the wild, social interactions, etc., that are playing out in the environment. Very large scales, often long time scales as well. In the middle is that space of connectomics, where we acquire and represent interactions among neurons or brain regions, either anatomical or physiological or functional, acquired with very many different measurement technologies, all the way from EM, electron microscopic reconstruction, all the way up to human neuroimaging. And those data sets are proliferating. There's more and more of these types of data out there. And we need a framework within which we can analyze and describe and model and understand these relational data. And that is given to us in part, in large part, I think, by network science. And in the past, a lot of network science methodology has been made available to us in neuroscience. Simple descriptive measures of how nodes and edges relate to each other in a topology. Very simple things like the node degree. How many connections are attached at each given element in your system, whether it's a neuron or a brain region. Or things like clustering coefficient. How cliquish is the neighborhood of a central node? How many of its neighbors are also friends of each other? Decomposing a graph into motifs or subgraphs and counting them, looking at the statistics of those to get a feeling for how the graph is actually built. The very important concept of path length and distance in a graph, which relates not to metric distance expressed in millimeters, but in number of steps that it takes to travel from a point A to a point B. The distance from A to B is the minimal distance, the minimal number of steps typically taken by, for example, as an agent or a message or information. It's a very important concept in the brain, and I'll come back to that throughout my talk. At the bottom, two other aspects that are very heavily researched in our field now. One is modularity. By that I don't mean Fodorian modules as in cognitive science, but simply a decomposition of a larger graph, a larger network, into components that are very densely connected internally, but somewhat more weakly connected amongst each other. Very important in that context are those nodes, those elements in the graph that are critical for keeping the whole system connected all together, for allowing information to flow between communities. And these are these so-called hub nodes, which are bridging between communities, allowing information to travel between them. Finally, the Rich Club is a concept that's coming also from the social sciences. You get the idea based on a name, which is simply an association, a dense connectivity between very highly connected parts of the network. In a social context, highly connected members of a social group often are also highly connected amongst each other, more so than expected by chance. It turns out that's an important concept also for the brain. It'll recur throughout my talk. One of the struggles that we have in our field in brain networks is that our data is extremely hard to acquire and to collect. And we have to spend, I would say, sometimes 80–90% of our time making sure our data is of high quality and that noise sources have been accounted for and have been eliminated. This is true for data sets from different species, whether it's the nematodes, the elegans, the rodent brain, the human brain, non-human primates, you name it. And extracting brain network data from anatomical or physiological observations is a time-consuming process. Entire careers are built on making methodology, testing methodology, just on this slide alone. Very briefly, and all too briefly, on the left side of this, what you're seeing is a rough workflow of how we construct structural networks, anatomical networks, which describe the associations between brain regions or neurons in anatomical terms. Quite often we end up in human imaging with a volume of the brain that we cut up into pieces, coherent brain regions, and we're estimating the density and strength and magnitude of the tracts, of the connections between these brain regions, often with the use of non-invasive neuroimaging information. We can also do histology, not in humans, but in other species. The end result of that is what we've called now, about 10 years ago, a connectome. A connectome, which is an ohm. Why? Because it's fundamental, it's universal, and it's comprehensive, a description of all the connections in a brain at some level of scale. Mathematically, it's a matrix. It's a matrix of pairwise relationships between nodes of the brain regions, and these nodes either have a connection or they don't, and that connection may also have a weight and a direction. That's a matrix, that's a network, a description of a network that we can now use for graph theory and network science analysis. On the right of this diagram is a whole different workflow. Now we are not measuring anatomy, we're measuring something about time series, about responses of the brain, either to stimuli or responses that occur spontaneously. That results in recordings of voltages or bold activations across time, wiggly lines over there, which we can process using time series analysis tools into, once again, a matrix of pairwise dependencies. This can be as simple as a cross-correlation, or it can be much more sophisticated. This matrix that you see on the right, we loosely call a functional network, a collection of all pairwise interactions, which can also be analyzed and modeled using the tools of network science. So we're getting a very nice approach here of using a coherent set of tools on both anatomical observations that are about structure and physiological observations that are about responses. And that's the framework within which now the field of brain connectivity and network neuroscience is operating. A few words about how these two modes are different from each other. I just want to underscore this because it's sometimes misunderstood. Both structure and function in this representation have network characteristics. So there are nodes and edges and relationships. And they may have topology, even multi-scale organization. There may be modules within modules, etc. But there's also important differences. Structure connectivity is physical, it's real. It's something that you can, technically speaking, poke with your finger. There's a synapse, there's a connection, there's a projection that you can estimate the magnitude of, but it's physical, it's real. It's often sparse. It's a sparse type of connectivity. And it may have complex properties such as biophysical properties, neurotransmitter properties, strength, effectiveness, etc. It changes across time, typically on a relatively slow time scale through development and across the lifespan, perhaps faster during plasticity. Functional connectivity is very different. It's the statistical estimate of some association between time series, as simple as a cross correlation. It is, for that reason, a very different type of object. Individual estimates of edges, for example, are not independent of each other. It is intrinsically dynamic. It changes across time much, much faster. We think in fMRI on the time scale of seconds, and then perhaps with EEG, MEG, and fast recordings, even faster than that. And there's an extremely large possible number of configurations that a brain network, a functional brain network, can go through on top of an ultimately fixed anatomical skeleton. So on the top, physical connections, often sparse, that we can estimate across the brain, but they're relatively fixed. On the bottom, something which is a very much fuller description of interactions. It changes much faster, and it has a virtually infinite number of configurations it can go through. Both of these are analyzed in the context of connectomics. Both of them form the basis of that new emerging field of connectomics. But I want to really emphasize that they are different ways of measuring, looking at the brain, and not all methods, not all network science tools, modeling tools out there are equally appropriate for both domains. So now a few words about connectomics across species. This is from a recent review article I had with Martin Mannenheuvel, a longtime collaborator, where we just sort of wanted to give a visual impression of the different species for which we now have connectome diagrams. This ranges from the nematodes, the elegans, with about 300 individual neurons, to very macro-scale descriptions of the human brain of hundreds of brain regions and their interactions with each other. And this sets up the possibility that we can actually compare. We can actually say, well, are there features that are different across different species, or are there features in this network architecture that are actually the same or have been conserved? And it turns out that I'll give you my bottom line, that a lot of what we see in brain architecture, anatomical structural connectivity, and how it's built across species is actually quite conserved. It's actually quite similar, whether you look at an organism as simple as C. elegans, or as one that, well, sophisticated is a tough word here, but that is as complex as the human brain. This is where the whole field got started in a sense, about 30 years ago now, with the largely manual reconstruction with EM micrographs of the individual neurons and their connectivity in the nematodes, C. elegans. Roughly 300 neurons and about 2,500, actually about 3,000 chemical synapses between them. And it continues to this day to be the only complete, fairly comprehensive connectome diagram at the cellular level of any organism. This was 30 years ago. I call it the moonshot sometimes because just like going to the moon, we haven't been back since then. It really hasn't been done, although we're going to see more cellular level connectomes, I think, in the next few years, especially from Drosophila. There's work that I hear is in the pipeline that's going to be much larger, give us much larger connection matrices than what we see here for another invertebrate. Now this matrix, this data set, which was acquired 30 years ago, is still being analyzed today. I still see papers coming out all the time, actually, that are looking at various aspects of this connectivity diagram. A few highlights recently, a few things that have been looked at with newer methodology. There's a modular organization. There is evidence for some neurons having a lot more connections than others. So not all neurons have the same number of connections in this nervous system. There are some that have a lot more, others have a lot less. And the neurons that are highly connected, that have a high degree, that have many connections with other parts of the brain, also tend to be densely connected amongst each other. They have a higher likelihood of making connections with each other than expected by chance, based on a null model. And that is the so-called Rich Club organization, the Rich Club feature I mentioned to you earlier. Going to another invertebrate species, very important one, Drosophila. This is work where I've collaborated with Ralph Greenspan at UCSD and Anshin Chiang at Tsingtao University in Taiwan. They constructed a mesoscale representation of the Drosophila brain on the basis of individual observations of fluorescently labeled neurons, mapping those neurons into a template space, common template space, and then reconstructing on the basis of that with tens of thousands of observations a mesoscale picture of what brain region is connected to what brain region, how strongly. And that's the little matrix you see on the lower left. It's also got modular organization above chance. It also has highly connected local processing units or brain regions in the Drosophila brain, and those LPUs are also connected amongst each other more densely than expected by chance. Common features that we have seen already in C. elegans. Going to the vertebrate brain, the mouse, this is the work coming from the Allen Institute now 3 years ago to very great fanfare. The version of the mouse connectome was published in Nature based on hundreds of injections in different animals, high throughput imaging of fluorescent label reconstruction, automated reconstruction, and then modeling, projecting it all down into a single matrix description of the mouse connectome. And this was then analyzed by Mika Rubinov and Ed Bullmore a year later, paper in PNAS. And one of the current features here, once again, are that there are some more well-connected regions. These well-connected regions tend to also be interconnected amongst each other in a rich club organization fashion. These are regions in the anterior cingulate, in the orbitofrontal cortex, in the thalamus, basal ganglia, etc. And you can see these in the matrix representation here. These are the red stripes that run vertically, horizontally, where the stripiness of the pattern indicates these are very highly connected regions that are also interconnected amongst each other. Going to another rodent species, the rat, this is a work that I've carried out now over the past 3-4 years with Larry Swanson at USC. Larry and his co-workers are hard at work scanning the existing literature on track tracing histology in the rat brain, and are building from that an annotated, correlated data set. And so what Larry does is scans the literature, extracts connection reports among brain regions based on published data, and then he builds connection matrices. Again, think of them as large Excel spreadsheets that are recording what's connected to what, lookup table. And our first foray into this was the cerebral cortex, single hemisphere, weighted connections, direct connections, because it's histology track tracing, and then looking at, again, the organizational features of this. Modularity is present, rich club organization is present, highly connected regions are present, and when we take those modules, we map them back onto the rat brain in a flat map representation, which is one of Larry's ways of representing connectivity and the architecture of the rat brain. We see that the modules we find are spatially contiguous. They involve regions that are next to each other on the cortical surface. Contiguity is a property that predicts being member of the same module. And that highlights the importance of spatial embedding in the way brain networks are laid out. The proximity of brain regions on the cortical surface is a strong predictor of whether they're connected or not, and in turn a predictor of whether they are members of the same community and perhaps functionally related. I'll come back to the importance of spatial embedding in just a second. More recently, Larry and Joel Hahn have expanded this map of the cerebral cortex to include both hemispheres and all the commissural connections between them, most of those traveling through the corpus callosum. And that allowed us to actually examine interhemispheric connections for the first time, really, systematically. I shouldn't say for the first time because the mouse brain, the Allen Institute mouse map had commissural connections also, and a paper on that has come out almost simultaneously with ours. Just a few of the rules we've been able to discover, some of the numbers here. This involved the annotation and curation of 32,000 connection reports from the literature, 77 cortical regions on both hemispheres, 144 in total. Data coverage 95%, so there is information on the presence or absence of connections in almost across the entire matrix. And we found that not all homotopic connections existed, so only about 2/3 of those homotopic connections between corresponding brain regions on the left and on the right side actually did exist. There was an even smaller density of heterotopic connections, only about 490 of those. And the commissurial connections were made across the two hemispheres were an exact subset of the association connections on the same side. So in other words, if there was a commissurial connection going across from the left to the right side of the brain, there also was, 100% of the time, a corresponding connection on the same side. Commissurial connections were in almost all cases weaker, and if you had a strong association connection on one side, a strong projection between two brain regions on the left or on the right side of the brain, you had a very high probability, 50% or so, of having a commissurial connection to the opposing hemisphere as well. See, that's some of the rules that coincidentally were also confirmed independently by Klaus Hilgetag and co-workers who looked at the mouse Allen data set. So that was very pleasing to us that these rules that we had extracted actually weren't restricted to the rat. This gives us some, we might speculate that commissurial connections might actually have similar organizational features across at least a few of the mammalian species, certainly maybe among rodents. Finally, to finish up the rat here, we also looked at, with Larry Swanson, at the basal ganglia, the cerebral nuclei, essentially striatum and pallidum. And those structures have very different gray matter and white matter architecture, and the connectivity pattern that we looked at with tools from modularity detection, for example, indicated to us, I'll skip forward here real quick, that the communities that we find in the basal ganglia have a much more asymmetric relationship with each other. There's much more, if module 3 projects to module 2 really strongly, there's only a very weak back projection between these two communities. This is something we don't see in cortex. Cortex is much, much more symmetrically organized. I don't want to go backwards now because then I have to pay some beers. This is a rule we had in a lab when Joaquin was around because he had a tendency to go backwards 10 slides. And I said, every time you go backwards, it's a beer now, okay? Anyway, we stopped counting at 237 or something. Anyway, so I don't want to go backwards, but it's actually a very different way in which different communities are associated with each other across these two major structures, major subdivisions of the brain. We are currently analyzing the rat brain, cerebral cortex, and cerebral nuclei on both hemispheres. So that matrix is starting to grow. It's now 244 regions, and we're starting to get data from Larry about the thalamus and the hypothalamus and the hippocampus, which is now being integrated into this. I think the vision is to gradually build from the existing anatomical literature out there a connection matrix of the entire rat brain, and look at this with our tools and modeling techniques. Finally, the non-human primate. In some ways, that's where I got started with the first and only data set in existence in 1991 from the work of Fellerman and Van Essen on interareal connections in the macaque visual cortex. That's been refined and expanded over the years into a database called COCOMAC. And a study we did a few years ago in the macaque cerebral cortex looked at the existence of highly connected regions once again and their interconnections. And you see up there the names. If you are familiar with your macaque neuroanatomy, you'll recognize these are prefrontal regions, cingulate regions, medial parietal regions, some temporal regions, widely dispersed, highly connected to the rest of the brain, and also densely interconnected amongst each other in a rich club organization. Here's where these regions are. Despite the fact that they are spatially distributed, remote from each other, they have connections that are quite dense, more so than we would expect based on some random null model. I'm going to skip the cellular analysis here and go straight to my sort of summary of this part of the talk. There's a number of features that perhaps you've noticed have recurred in my presentation. Almost all data sets that have been looked at have some admixture of highly connected brain regions. This is a non-trivial observation. You might expect that one brain region is like any other brain region. It's got inputs and outputs and about equally many. But that's not true. There are some brain regions that have a lot more connections, a lot more diverse connections, widespread connections than others. And those are putative hub regions that are important for integrating information, for dispersing information, and they'll make an appearance a little later in the human brain as well, when I talk about that. We also have, for every brain region we ever looked at, we have a specific connection profile. In some ways, if you think about a network, your status in the network, your contribution to the network is dependent on your connections. This is a very different way of looking at neural processing or at cognitive, cognitive function in a system such as the human brain. We are not looking so much at what's happening within each region. We're looking more at what's happening between regions and the capacities that arise based on their interconnections. And every brain we've looked at so far, every node, every region has its own specific connection profile, which is actually quite predictive of its functional specialization of its capacities. We find interconnected modules, and we also find that highly connected parts of the brain tend to aggregate together into a core, sometimes called a core, sometimes a rich club, sometimes a clique. There's different terminologies out there, but basically indicating the same kind of organizational feature, which is that there's some sort of central processing unit, a CPU, if you wish, where information from individual modules comes together and is shared. Why are there these common features? Many of them can be explained on the basis of spatial embedding. The brain is a spatially embedded system. It's a geometric object. This is a trivial fact, right? We don't think of it that way sometimes, but the fact that the brain has to exist in space is actually very important with respect to how it's connected. Why? Because connections incur cost. Connections are little cylinders that have to be biologically maintained. They take up energy, they take up space and volume. And so there is a premium on having a connection. There's a cost associated with it. That is one of the main driving factors already recognized by Ramon y Cajal, actually. We have, working with at Bulmor some years ago now, we came up with a sort of an idea or a framework for how to understand the features of brain connectivity that are actually shared across species. That has to do with the idea of a trade-off. On the one side, the brain wants to be cheap, has to be cheap. It has to fit in between your ears. If it doesn't fit between your ears, you're not going to have any offspring. Sorry. And that means this is not going to happen, okay? So it has to fit in here. That's number one. It seems like a trivial point, but it's actually of overwhelming importance. It has to be metabolically sustainable. The cost of signaling has to fit within the overall energy budget of the organism, and it turns out the brain is pushing the boundaries. 20% of our energy is devoted to 2% of our body mass. And so it wants to be really cheap as possible. At the same time, a really cheap brain might look like the one on the left, which is that only nearest neighbor brain regions or neurons are connected. That's cheap, but it's also terribly inefficient. If you need to share information from the back of the brain to the front, well, good luck with that, because you're going to have to go through 1, 2, 3, 4, I don't know how many steps we've got. Okay, so that's not good from the point of view of functioning. A random network, on the other side, can share information extremely quickly, but being random, it has no organization to it. The nodes have lost their specialization. They have lost their fingerprints. That's not a good brain. It's cheap. It's efficient, but you really can't do anything with it. Plus, it's super expensive. Somebody once computed, I still want to reconstruct this at some point, that with all the neurons we have in our head, if those neurons were randomly connected, the volume of the connectivity would blow the brain up to a size of something like 10 miles across. It's an absurd number, but so there's got to be a bug in that calculation. But just suffice to say, it's probably going to be not going to fit anymore, right? It's actually very wasteful. So that isn't going to be helpful at all. So the idea is we have a trade-off. It's like an engineering concept, right, or an economic concept. We want to optimize two things, efficiency and cost. We have to settle on a compromise. Most of the time, the brain is very cost efficient. And then some of the time, features of brain connectivity appear, such as rich club organization, that are actually quite costly because you're connecting widely dispersed regions that cost you a lot in terms of volume. But you need to do it because you have to integrate information. And you have to guide behavior and cognition that way. So that's why we think that's a useful framework to pursue. It is something that we've looked at in the context of morphospaces, an idea that's actually of interest to people here, and an idea that we continue to pursue in cross-species comparisons as well. Okay, what about the human brain? I've only talked about animal brains so far. When I went to high school and then to university in Germany, I was already interested in brain, but the only thing I could find about brain anatomy was pictures like this in medical textbooks. This is actually a photograph of a microdissected brain of a dead person, obviously, and in literally, it gives you a nice visual impression of some of the so-called white matter tracts in the interior of the brain. Myelinated axon bundles that connect remote brain regions with each other. And now you can see the disadvantage of doing this is not going to be a person you can bring back to the lab to ask them questions about their cognition and behavior and test them again. No, that's a one-way road right here. And you can't do this on thousands of specimens either. And you have a biased sample when you do postmortem brains. Not a good way of getting an idea of how brain structure varies across the population, etc. Well, enter the arrival of diffusion imaging and tractography. These images here that are now very commonly seen, including on one of your newsletters on the front page I saw today, were unknown 10 years ago, basically. Okay, we had no, just about 10 years ago, we had essentially no comprehensive information about human brain connectivity, anatomical brain connectivity. Also, resting state was something that crazy people did, like we. So 10 years ago there was nothing, okay, and we had the technique was just being developed. It continues to be refined. It is rather tricky. And on both on the acquisition side as well as on the reconstruction side. This is a picture that's a computational model. This is not a photograph, obviously. So you have to appreciate that what one does here is infer the most likely layout of white matter connections based on a very obscure signal you get from the inside of the brain while the person is alive. So it's very different than histological or anatomical direct observation. So there's many caveats to this technology. Nevertheless, about 10 years ago, I got together with Patrick Hagmann, my longtime colleague and friend from Switzerland, and he had been refining this technology. We had been working on networks with animal data, and we decided to combine our expertise, unfunded, by the way, completely unfunded, and constructed the first diagram, such as this one here, a matrix of connectivity of 1,998 parcels or brain regions across the cerebral cortex that describes the pairwise anatomical relations between these regions. And I say it's an early draft because many such maps have since been constructed, many of higher quality than this, and it's an ongoing project of further refining the accuracy and the validity of these types of reconstructions. The results we came up with in our 10-year-old paper echo some of the things I mentioned earlier. This is not a chronological talk, actually. A lot of this work here on this slide preceded what happened earlier in the talk. Okay, in some ways, we got started on the human brain and worked backwards to animal species. Unique fingerprints, broad degree distributions, which means an admixture of high degree nodes. Some brain regions are more connected than others. Clustering, modules, and a highly connected core. We called it a structural core at the time. Later on, we called it a rich club. These are descriptive markers of brain architecture in the human brain that can be quantified, that can be compared across individuals, that can be modeled. And there's a lot of descriptive analysis that has happened in the last decade or so, But an increasingly more important method now is to construct generative models, to construct models that that ask questions about what are the principles behind, why does connectivity look the way it does? What generative mechanism can explain that? And a couple of the recent papers, one from our lab, one from Mika Rubinov, that have been examining this. It's an important issue because if all you do is describe features of connectivity, you can be fooled into thinking something is highly adaptive, significant, important, but it actually turns out that it can't be any other way. It turns out that your brain has to look this way because it's spatially embedded or some other factor plays a role. But then it can't be adaptive, right? If it has to be that, this is the old spandrel argument from evolutionary theory. So that's why generative models are very important to sharpen our understanding of where the features of connectivity we actually observe, where they actually come from. I want to revisit just very briefly the Rich Club with you, just to hammer that home one more time. Here's how this works on the left. Left diagram is a schematic of a network with modules, communities that are more densely connected amongst each other, but weakly between. We have these highly connected nodes that connect them to each other, those black nodes you see there. In the middle, all I've done is I've added a few connections between these highly connected nodes. Now I have dense connectivity. You can perhaps see right away why that's beneficial. I now have an infrastructure in place in this network where information can be sampled from different communities that are perhaps functionally specialized to do very different things, vision, audition, some other sensation, what have you. And I can now bring this information together in this central red-colored Rich Club network. So we were interested in understanding, based on this intuitive notion that the Rich Club would be a great sort of infrastructure feature of the human brain, we wanted to see whether it's actually there. And that work started with my collaboration with Martin Vannenheuvel. And long story short, analyzing a set of human subjects at different resolution levels, yes, we find evidence for more dense connections among highly connected nodes than we would expect based on a, I think, reasonably conservative null model. And furthermore, if we ask the question, if we want to communicate from any point to any other point in this network, how often do we have to go through the Rich Club? It turns out 90% of the time. Any pair of nodes in the brain, you want to send a message on the shortest path possible, the most efficient way of sending the message, you have to access that central red blob that you saw earlier, despite the fact that that's only about 10% of the brain. I don't want to start another meme here about we're only using 10% of our brain, okay? Do not get me wrong. This is not what I'm saying. But there is a tendency of that central core, it sort of absorbs traffic. Traffic naturally goes into it and comes back out if you want to communicate across different parts of the brain. At least in a model, that's the case. Finally, if we lesion this central core of the brain, it has disproportionate effects on communication, potential communication, on the coherence of the network. And that, in turn, is of crucial interest to people who have clinical translational interests. I think one of the big payoffs of Connectomics has been its application, mostly in human neuroimaging, to disorders of various kinds. I just throw this one slide out here because there's a whole separate talk to be given, or a whole course to be given about the importance of hub regions and interconnections among them for human brain and mental disorders. The diagram here on the left is from meta-analysis of over 20,000 structural scans from people with various conditions. I think 30 or so disorders, everything from autism to schizophrenia to depressive disorder to epilepsy to panic disorder, chronic pain, dementia, you name it. It's on that list. And the question that was asked here by Nick Crossley and Ed Bullmore in this meta-analysis was, in these people, where was evidence for atrophy, for structural damage to the brain? What were those regions, and how well connected were them? Where were these regions? It turns out that there's a statistically significant bias in the direction of most disorders are associated with structural damage to nodes that have high degree and are therefore putative hubs in the brain. There's a plethora of evidence, not just this meta-analysis, but a plethora of studies now that point to brain disorders and mental disorders manifesting predominantly through some disruption of highly connected nodes. You might say, if you think about what we think these nodes might be doing, this seems obvious. There's, just by analogy, think about the air transportation system. You know, if Chicago shuts down, it's bad for many people in the air transportation system. If Indianapolis shuts down, not so much. Sorry to say it, but we're not a hub. Although we have now one flight to Paris. Nobody. It doesn't disrupt the system as much as if a hub shuts down. This is perhaps a basic insight, but I would say that was not at all part of the conversation even just 5-10 years ago. So there's a lot of interesting leads now. It turns out hubs metabolize glucose differently. It turns out that when we stimulate them, perturbations travel differently across the brain. There's a lot of interesting, they have different gene expression patterns. There's a lot of sort of systems level peculiarities about them that place them high on the list of interest in clinical translational studies now. Finally, functional connectivity. I've talked a lot about anatomy so far, and perhaps because I'm very fond of anatomy, but ultimately we want to know how the brain responds to stimuli, or perhaps how it becomes spontaneously active. That's a movie on the right that shows you a projection of fMRI bold responses onto the surface of a brain in a person who does nothing in the scanner, so-called resting state. Just like the fellow before, please ignore me. The instruction to people going into this type of experiment is don't think of anything in particular. It's a task to me, but okay. And so resting state is essentially spontaneous, self-generated brain activity. And it looks like this. The amplitude of these bold responses, by the way, is as high as anything you do during a task. These highly processed sort of task, bold pictures we are very familiar with the hotspots, the amplitude of those peaks are actually about the same as this. So if I threw a task in here, you wouldn't notice it. It would become part of the ripple pattern of this waveform unfolding across the surface, just as an aside. So it's not noise. It's not a little bit of background hiss, in the microphone. It's actually using up 60 to 80% of all brain metabolism goes into resting state activity. And there are now such things as resting state networks that have names, such as default mode, saliency, visual, etc. How do we construct those? We take frames from a movie like this, chop it up into frames, do a cross correlation, cluster it to get coherent blocks of brain regions that are coherently active. And then we project it back onto the cortical surface, and we give them names. Now we know what things do. Once we give it a name, we know what it does, and it's all over. Default mode, visual network, frontoparietal. These are now names that are frequently used in the neurocognitive literature as building blocks of cognitive processes. This is a very much accepted way of mapping brain networks now. You can reproduce this very easily in any cohort out there. It's a very highly reproducible feature of brain network organization, now functional networks, mind you. So how can we model this? We actually started in a lab over 10 years ago now, trying to simulate fMRI responses in a computer using computational neuroscience tools. We started with a coupling matrix. This is a connectome anatomical coupling matrix, in this case coming from the macaque brain, because that's where the whole thing got started in the 90s. And each node in this matrix is now given some biophysical properties, excitatory and inhibitory currents, some time constants. So in other words, this is now a biophysical so-called neural mass that can behave, it can oscillate. Once we take a bunch of these equations at the bottom, couple them up with those coupling terms at the top, essentially we're getting now a coupled system of nonlinear differential equations. This is physics. Engineering. This is engineering, right? We can numerically simulate this in a running it, going, piping it through an ODE solver, and we get time series. We can then, perversely perhaps, take these millisecond time series and pass them through a filter that's like an fMRI response. And then we can get cross correlations from that, a functional connectivity matrix. We did this work back with Chris Honey, 2006, 2007, before resting state was actually accepted in the community as an honorable thing to do. And the great thing about a computational model like this is you can manipulate the model. You can perturb the matrix. You can make lesions. You can take out a brain region and see what the rest of the brain is doing. You can add a connection if you want. You can make connections weaker and stronger. You can provide inputs that might simulate task conditions. And you get variations of this connection matrix on the right, which you can then compare to empirical data. In our case, we had no fMRI resting state data back in 2007, but years later, Yasushi Miyashita and Tokyo contacted us wanting to compare our simulation to empirical data he had acquired, and to our great happiness, the relationship between the two was, I would say, robust. Given that we did no data fitting at all, given how simple the model was, no neurons, just neural masses, 47 of them, with a matrix that had many gaps and holes, we were actually quite pleased with the amount of agreement between empirical and model data. Now, one way that we can pursue this further is we make more detailed models. We make spiking neuron models. We have put in layers. We put in more brain regions. We put in inhibitory neurons. Who knows what? But partly as a result of Joaquin's interest in this, we actually, there you are, we actually went a different route towards much more simple models, which are in fact analytic. You solve them directly on the connectome matrix. And this was a multilinear model of 4 different graph descriptors that relate to communication. A couple of them were path length, and another one was the so-called search information, which is the degree to which a path is hidden from discovery by a random walker. Turns out that search information becomes more, it's growing. You need more and more information. The more you have high-degree nodes along the way. If you travel through Times Square, your chance of getting lost are much higher than if you just walk down a village which has a single main road and that's it. There are no branchings. And as Joaquin elegantly showed in this wonderful paper that came out now 3 years ago, we can do really well predicting functional connectivity with a simple linear combination of graph descriptors related to communication. This fancy, I really like this diamond graph. On the left side of it, we have an fMRI bold response cross-correlation matrix coming from resting state individuals. The right part of the diamond is the model prediction based on the structural connections of those same individuals, based on the anatomy, which you don't see here. And despite the simplicity of the model and the fact that it runs in MATLAB in under a second, we can predict this very, very well, fitting only 4 parameters. And as you can see, it works quite well across different parts of the brain as well. That whole line of work kind of triggered in the lab a whole interest in communication and communication dynamics that goes on to the present day. We kept thinking about, and we had many sessions on the whiteboard in the lab back when Joaquin was around, about how communication can unfold on a network, and especially in a brain network. Think of the topology of an anatomical network as constraining the way in which information can disperse and flow. Obviously, it can't flow through the ether or the air, but it has to go through these paths. And while these connections are quite sparse, there's many, astronomically many paths in these networks. I mean, the numbers are really staggering. And functionally important communication arguably involves communication events, signaling events, action potentials traveling through axons and pathways. And a lot of these connections, because the network is sparse, a lot of these events have to occur through indirect pathways. So it has to go from A to B to C to D. It doesn't go from A to D directly if A and D are not connected. Just as a back of the envelope calculation, we have about, give or take 80 to 100 billion neurons. And that would be a matrix now. Think of an Excel spreadsheet with 100 billion rows and columns, and you're making entries as to which of these neurons are connected. It turns out only 1 in a million or 1 in 10 million of all the entries in that matrix would be filled. So the likelihood of having two neurons that are connected, if you pull them at random from the brain, is 1 in a million. Which it stands to reason that a lot of communication has to occur, certainly at the level of individual neurons, has to occur across indirect pathways. Otherwise, the brain simply could not come up with an integrated response to anything. And the concepts that have been most influential in the past have been shortest path. The most efficient way of passing from A to B is to, let's say, a minimal number of 2 steps. And so-called efficiency, which is the inverse of the path length across all pairs of nodes in a network. And those have been the dominant concepts in our field. Most papers that deal with efficient information transfer so far make reference to these two concepts. But there's a problem with what we might call the classical view. Shortest paths are great because them being short, they're efficient, there's not much noise corruption, delays are minimized, information arrives fast, travels fast. But how do you find it? Put yourselves into the shoes of a little neuron in your brain, if it has shoes or feet for that matter. Okay? It doesn't know anything. It gets an input now and then or some inputs, it produces a spike, that's it. But it has no map of where information should travel. It has no concept, obviously, of the topology of the brain network as a whole. When we plot shortest paths in a given data set, we use powerful algorithms that have names like the Dijkstra algorithm that allow us to find these paths efficiently and fast. But in the brain, nobody can do that. Those little neurons in the little shoes, they can't do this. So how does information know? How do neurons know how information should flow efficiently, if it in fact does flow efficiently? So that's a problem of global knowledge, which we supply when we do computational analysis, but neurons don't have. So we have sort of some alternatives. First of all, there's many, many alternative paths, and believe me, the numbers are in fact staggeringly large, that are almost as short as the shortest path, but not quite. And that's work that Andrea Arena-Königsberger has done a couple of years ago now, and the concept that she's coined is path ensembles. These are ensembles, sets of paths that are almost equally short, but actually involve a lot of the infrastructure of the brain. And another thing we have to think about is that not all paths are equally likely to be functionally meaningful. Efficiency says it's the inverse of the path length over all pairs of potential interactions, but nobody says that all of them are equally important or likely to occur or meaningful. So we have to kind of think of communication in different ways. We are now having a framework in mind in our lab where we're thinking of communication events as being real drivers of functional connectivity. This is sort of a new concept. Essentially, it says functional connectivity, the time series fluctuations we observe after we record from neurons or brain regions, are the result of lots and lots and lots of communication events, signaling events that occur underneath. We actually don't see those directly. We have a review coming out in just next week in Nature Reviews Neuroscience that articulates that theory or that framework in a bit more detail. But just to give you the rough idea. So I've talked a lot about structural connectivity, anatomy, physical wiring. That's the infrastructure. Those are the highways, if you wish, that are costly because they are built from asphalt, and require to be maintained, etc. They are sparse, and they are the connectome. Then we have on top of that lots of communication events. Neurons are spiking. Brain regions are sending out compressed and coded signals or messages down their axonal pathways. Those are spreading on top of the connectome, on top of that structural network, and constitute communication events or communication dynamics, communication events that unfold in time. And then as a result of all this happening, we see that time courses, responses of neurons and brain regions have these characteristic deviations up and down, have certain characteristic patterns to them. And we capture those with functional connectivity. The point is that we can access this because we can record from the brain and do things like cross correlations. We can also access that. It's hard to do, but we can measure the connectome better and better every year. We have no access to this. We actually don't know how to observe individual signaling events unfolding across time in the brain in real time as the brain is responding. And I actually have a hunch that this is a different mode of connectivity that perhaps is akin to what Carl Friston and others have been writing about in terms of effective connectivity, causal interactions among neural elements that are driving behavioral responses and cognition. And we have, in the context of this review that will be out next week, we've tried to sort of put all these different communication models that are out there, including routing, which is shortest path communication, and also random walks, which is diffusion, sort of on a spectrum of the amount of information that's needed to actually make this scheme work. Here, you have to have a lot of information, a map. This is like FedEx, sending you a package, and the package is not. They don't send it to a random, or at least they shouldn't, send it to a random, office, and then that office throws a die and sends it off to another random office. You would never get your package, right? And sometimes that does happen, apparently. But that would be a random walk. It's very inefficient, but you also need almost no information at all. So there's an informational cost to communication that has been underappreciated. We have been, in our thinking in the field, largely on this side, but we have ignored the fact that we can't get this, or the neurons can't get this. But if neurons work like this, it's extremely wasteful. And in fact, the whole notion of messaging or information transfer is lost. It's more like broadcasting. It's more like shouting. And this is one thing. Not many things, make me wake up at night, but this is one of them. Here's something that has occupied me over the years. I actually started my career as a biochemist, so I was down here somewhere. I worked my way up the scale. Now I'm all the way up there. And as someone who has brought interest and has been involved in many, many projects over my career, it has occurred to me that we really do have networks, operating systems at very different levels of scale and organization in the brain. Almost any process that we're excited about or interested in that involve systems in neuroscience involves networks on multiple levels of organization, interacting proteins inside cells that interact to form tissues and circuits, and ultimately whole brain systems that support and enable cognition and behavior. And to have a deeper understanding of what goes on here at an integrative level, we need to do better in neuroscience to connect these levels up with each other. We have many specialists who are down here, we have many specialists who are up there, but we don't have a lot of concepts and models and theories that actually can effectively link these different levels together. And in part to promote that, and since I have nothing else to do with my life, I started a journal last year called Network Neuroscience, published by MIT Press, open access obviously, that is devoted to, first of all, network science and neuroscience interactions and intersections, both empirical and computational work, but then also hopefully can pave the way to a greater appreciation of the network basis of brain function across different levels of organization. And with that, I thank you again for the invitation, and I'm very happy to be here. I hope I'm back soon. Thank you very much.